In previous lessons, we learned about allocation, that a certain set of cells will be allocated to forming a memory trace of a learning event, and the allocation happens both in the hippocampus and in the cortex, and the effect of system consolidation is to strengthen the cortical memory trace. So the interaction between the hippocampus and the cortex over time allows the memory to be more predominantly stored in the cortex and accessible by cortical retrieval systems. In this lesson, we'll continue to examine these concepts and tell a more detailed story. First, we'll tackle the question, why is the hippocampus such an important structure in memory processing? Well, it has to do with the kinds of inputs that the hippocampus receives. The hippocampus receives information from all of the major sensory and perceptual areas. Visual cortex in the back of the brain, somatosensory cortex, the touch cortex here, auditory cortex here. All of these areas send information down into the hippocampus. And that makes it possible for the hippocampus to link those perceptual qualities of an experience. Now, the first stage of a learning event, psychologists call the encoding stage. So here we have the surface of the brain, and let's imagine we're in a classroom, and we're using microscopes, and we're observing amoeba and paramecia under the microscope. Meanwhile, a teacher might be saying something like, amoeba extends pseudopods to engulf the prey. So the different sensory and perceptual areas of the brain are processing the different aspects of the experience. And within each region of the brain there, a subset of those neurons will be responding to the specific stimuli, and they will be allocated to storing a memory of that aspect of the experience. So the memory of what amoeba and paramecia look like will be stored in visual areas. The sound of the word amoeba will be stored in auditory areas. What it feels like to handle the microscope will be stored in the touch areas of the cortex, etc. Now, all of these perceptual regions send information down into the hippocampus. So we would say there's a convergence of that information in the hippocampus. And within the hippocampus, then, here's a magnification of the hippocampus, which really is underneath the temporal cortex. A subset of hippocampus neurons respond to the information from the perceptual areas, and some of these neurons will be allocated to forming a memory trace of the episode. And for simplicity, we sort of identified the green one here as being activated by the sound of the verbal descriptions in the learning environment, the blue one by the feel of the microscope, and the red neuron by the sight of the amoeba and paramecia, etc. So a memory trace will be formed in the hippocampus. Now, in keeping with our diagrams in previous lessons, we're going to now tell the story using a brain that looks like this. The circles would be neurons or brain cells, and they are here in the visual cortex and the touch cortex and auditory cortex, and here we have the hippocampus underneath the temporal lobe, and we have hippocampal neurons here. The first stage then, as we've just seen, is an allocation stage where a certain set of neurons in each of the perceptual regions will be activated by the learning experience and then allocated to storing a memory trace of that learning experience. The perceptual regions will send information by way of axons to make synapses on neurons in the hippocampus. So we have a convergence of information in the hippocampus. The unique learning episode activates a subset of hippocampal neurons. That's an allocation process. And scientists suspect that the most excitable neurons at the time of the learning are preferentially activated. But the nature of the learning event may influence which neurons are activated as well. So for this given learning event, where microscopes are being used and amoeba and paramecia are being witnessed and we're hearing things about amoeba, this particular set of hippocampal neurons becomes activated. When those hippocampal neurons are activated, the simultaneous activity of those hippocampal neurons causes them to undergo a growth process that strengthens their connections to each other. And this is where neuroplasticity comes into play. We'll say more about that in future lessons. So neuroplasticity, that's a growth process that will strengthen the connections, strengthen the communication between those activated neurons. That takes place in the hippocampus as a result of the learning event. And thus we call this a strong memory trace, otherwise known as an engram. So neuroplasticity is the key to setting up a population of cells here that represents a memory trace of the learning event. Now, the memory trace in the hippocampus is like an index. It is connected to the cortical memory traces of the event, and the index represents the pattern of cortical activity that occurred during the learning episode. So this memory trace in the hippocampus is connected to the memory traces in the cortex. After all, the cortex, these regions were sending the information down into the hippocampus in the first place. So we want to think of this index as sort of representing a pattern of neural activity out in the cortex. However, the cortical memory traces are weak, meaning the connectivity between the cortical neurons is not as strong as those in the hippocampus. And we're indicating this with dashed lines between the cells involved in the memory trace in each region of the cortex. So the hippocampal memory trace is strong, indicated by solid lines here, but the cortical memory traces are weak. Now, at this early stage, perhaps the next day, if somebody said the word amoeba and asked you to kind of retrieve information that you have about amoeba, that's a retrieval cue. And the idea is that after we've learned about amoeba the day before, the retrieval cue is going to activate part of the cortical memory trace. Remember, the cortical memory trace included all of these regions of the cortex here, but the cue, if I just say the word amoeba, that's an auditory cue. So auditory cortex will activate those cells that store the memory trace for the sound of the word amoeba. And they will then activate the hippocampus, just as we saw the day before during learning. So those cortical neurons activate part of the hippocampal trace. So you see the green cells being activated here. But notice then, because hippocampal neurons in the memory trace are strongly linked as a result of neuroplasticity, activation of one neuron reactivates the entire memory trace. And this is called pattern completion. The activated index can be used to reactivate the cortical memory traces. So notice initially the green cell here was activated, but because of strong connections to the red and the blue cell, the entire hippocampal memory trace can be reactivated. And scientists call this pattern completion. Now, when the hippocampal memory trace then reactivates the cortical memory traces, this is what we mean by memory retrieval. At this point, we would experience the retrieval of the information from the day before, what an amoeba looks like. Maybe we learn some facts about the amoeba. We might be remembering the feel of the microscope, etc. Now, for a period of time after learning, the hippocampal index is required to reactivate the cortical memory traces. And this is because the cortical memory traces are weak. So if we're going to access these cortical memory traces, we need the hippocampus memory trace to be first activated so that then it, through its connections back to the cortex, can reactivate the cortical representation of the memory. So early on, we need the hippocampus to retrieve information stored in the cortex. And that's because these memory traces in the cortex initially are weak, as shown by the dashed lines. However, system consolidation has the effect of strengthening the cortical memory traces. And during consolidation, the communication between the hippocampus and the cortex is similar to what happens during memory retrieval. What scientists think is going on, and we'll see this in the next lesson, is that the hippocampal memory trace gets reactivated in a way that it can then signal the cortex to strengthen just those connections between the relevant neurons, the neurons that were allocated to storing the memory trace for that aspect of the experience. So system consolidation then is this communication between the hippocampus and the cortex, and the nature of that communication strengthens the cortical memory traces for that learning event. And this shares something with just sort of retrieving the memory, because again, early on, to retrieve information, we have to first activate the hippocampal memory trace, which then subsequently activates the cortical memory traces. Well, scientists think system consolidation is doing this very thing. It's reactivating the hippocampal memory trace and using that activation to strengthen the cortical memory traces. Now, the result of system consolidation is a network of strong cortical memory traces. Memory traces are linked in semantic memory so that partial activation of the network can reactivate the entire network. So notice then we still have our strong memory trace in the hippocampus, but after consolidation, the cortical memory traces are now stronger. Notice the solid lines between the cells. And again, the idea is for that learning event where we were looking at amoeba and paramecium under the microscope, the visual appearance of amoeba and paramecium would be stored in cortical memory traces in the visual part of the brain. What the microscope feels like would be stored in the touch part of the brain. The sound of the word amoeba and paramecia would be stored in auditory regions, and all of these cortical memory traces can be linked together. And scientists start to then describe this kind of network. This would be the concept of an amoeba. So if I just hear the word amoeba, I can activate the concept and then reactivate what an amoeba looks like. Or if I see an amoeba, that will activate the concept, and then I can activate the word amoeba or other facts about the amoeba. So this is what it means to have semantic memory, so basic information about the world. We are setting up memory traces in distributed regions of the brain, but linking them together so we can access all the different kinds of information associated with one concept. Now, after sufficient system consolidation, then, the hippocampal index is no longer needed to reactivate the cortical memory traces. Frontal cortex retrieval systems can activate information stored in semantic memory. So at this stage then, after successful system consolidation, the cortical memory traces are strong enough that they can be reactivated by frontal cortex systems without the need of the hippocampus. And so we saw this with patients like HM that had access to semantic memories even though he didn't have his hippocampus. Now, of course, we continue to have, in intact people, we do have the hippocampus, but we don't necessarily need that index in the hippocampus to retrieve the information that was stored in the cortex. In fact, there are good reasons to do system consolidation because changes are occurring in the hippocampus that will wipe away memory traces. Hippocampal memory traces are vulnerable to interference and disruption, and we'll see why that is in future lessons. But system consolidation then is going to be setting up strong, stable, reliable memory traces in the cortex, in part because the memory traces in the hippocampus, while they were strong early on, they're temporary. There are forces that are going to be disrupting and weakening and eliminating and clearing memories out of the hippocampal system.